Hello everyone, I'm Suryan. Today I'm going to explain about what is bacteriocins, its characteristics and its types and what are these application for bacteriocins. So first definition. Bacteriocins are ribosomally synthesized antimicrobial peptides or proteins produced by bacteria that inhibit or kill closely related or sometimes broader group of bacteria. Unlike antibiotics, which are secondary metabolites, bacteriocins are gene-encoded peptides produced during normal bacterial growth. History. The first bacteriocin discovered was colicin, produced by E. coli in 1925 by Henri Gretria. Next, the characteristics of bacteriocins. Bacteriocins are protein or peptide in nature. It's ribosomally synthesized, usually narrow spectrum, active against closely related species. Heat stable, many are stable up to 100 degrees Celsius, activate at low concentration, degraded by proteolytic enzymes like trypsin, pepsin. Now let's see about bacteriocin class. From gram-positive bacteria, most studied from lactobacillus or bacillus species. Class 1 bacteriocins are antibiotics, small peptides post-transnationally modified, contain unusual amino acids, lanthionine, example niacin. Class 2 bacteriocins. It's non-lanthionine peptides, small, heat stable, no unusual amino acids, example pyriocin. Class 3 large proteins like bacteriocins, heat labile, 30 kDa size. From gram-negative bacteria, mainly from Escherichia coli, colicins, large proteins, microcins, small proteins. Next, bacteriocins produced by lactic acid bacteria. Lactobacillus curvatus, LAB3H, produce bacteriocin lab3H. Pediococcus acidolactici, PAC10 cultivated in MRS broth, produce Pediosin PA1 bacteriocin. Pediococcus damnosus NCFB 1832 isolated from beer, produce Pediosin PD1 bacteriocin. Lactococcus lactis ATCC 11454 produce niacin bacteriocin. Pediococcus pentosus NCDC35 produce Pediosin NCD35. Lactobacillus paracasi SG1 produce paracasine SG1. The mode of action of bacteriocin. Bacteriocin kill bacteria by pore formation. They bind to cell membrane, create pores, leak of ions, potassium and hydrogen, cause cell death to the bacteria. Example niacin. Cell wall synthesis inhibition. Bind to lipid to block peptidoglycan synthesis, cause cell death to the bacteria. Nucleus activities. Some degrade DNA, RNA. Protein synthesis inhibition. Factor affecting production. pH. Optimal pH was 6 to 7. Temperature 30 to 30 degrees 37 degree common. Carbon source, nitrogen source, salt concentration, aeration. These factors affect production of bacteriocin. Applications. Food preservation. Niacin is widely used in dairy products, used against Listeria monocytogenes, Clostridium botulinum. Medical application. Alternative to antibiotics, active against multi-drug resistant bacteria, wound healing probiotics. Now let's see about bacteriocin clinical applications. Pediosin PA1 produced by Pediococcus acidilactici UL5 It targets Listeria monocytogenes. Its application was prevent intestinal infection in mouse model. Bacteriocin CC2 produced by Enterococcus faecinum DSM 2477. Its target organism was Streptococcus mutans. Prevent oral cavity. Bacteriocin AS48. The producing bacteria was Enterococcus faecalis UGRA10. A target organism was Propionic bacterium acans. Is used to treatment of dermatological diseases. Plantaricin EF. It producing microorganism was Lactobacillus plantarum. Its target organism was Mucispirillum and Parabacteriocytes. Its clinical application was treatment of acute inflammatory bowel disease. Enterocin DD14. Its producing bacteria was Enterococcus faecalis 14. Its target organism was methicillin resistant Staphylococcus aureus MRSA. Its application was treatment of multi-drug resistant bacteria. So different between bacteriocin and antibiotics. Antibiotics was secondary metabolites, whereas bacteriocin ribosomal. Antibiotics are broad spectrum, but bacteriocins are narrow spectrum. Antibiotics have high toxicity, whereas bacteriocins have low toxicity. Advantage of bacteriocin: it's safe to use, biodegradable, less resistant development, specific targeting. Limitation of bacteriocin: narrow spectrum, protein sensitive, expensive purification. Thank you.